Hello, everyone. I'm Dr. Terry Constantine, the CEO of Brain Research Foundation, and it's my pleasure to introduce Dr. Clarissa Waits from Columbia University. Dr. Waits was the recipient of the Jacob Jameson Huzenis Memorial Seed Grant, and she's going to tell us about her research, how exposure to chronic stress is a risk factor for developing major depression as well as Alzheimer's disease. We will save some time at the end of the talk for questions. Now, let me turn it over to Dr. Waits.
Great. Thank you very much, Terry. So I will just jump in here, I guess. So I'm talking today, like Terry said, about how chronic stress alters the brain, which is maybe sort of a timely topic for us right now. I probably don't really need to introduce the concept of stress, particularly in the current environment. It's very likely, I think, that all of us have felt stress for days or even weeks or months at some point in our lives. I'll give kind of a quick refresher for those of you who are a little rusty on your high school biology. So our body's stress response system is called the hypothalamic pituitary adrenal axis, or HPA axis. In response to stressful stimuli, the brain releases a series of hormones, which basically culminates in the release of glucocorticoids, which are the major stress hormones from the adrenal gland. And glucocorticoids, in turn, contribute to this fight or flight response that we all are familiar with from when we're nervous about taking a test or a job interview or, giving a talk that's live streamed on YouTube. So we get things like dry mouth and pupil dilation and racing heart and increased sweating as a result of this glucocorticoid release. And the stress response is actually highly adaptive. There are times when, our lives depend on it, but not when it's chronic. And this picture, I think, kind of nicely indicates how chronic stress has these profoundly negative effects on the brain. For instance, stress is a known cause of depression and anxiety, and these are really common disorders affecting about 25% of the US population. And there's many self-help books on the link between these conditions, as you can see here. Stress is also linked to Alzheimer's disease, which is the most prevalent neurodegenerative disease, which affects about 6 million Americans. And this is really a public health crisis because these numbers are also projected to triple by 2050. And as this illustration shows here, chronic stress is basically a driver of Alzheimer's disease. And then the symptoms of Alzheimer's disease in turn cause more stress. And so this leads to this vicious cycle. Interestingly, depression, which, as I showed in my previous slide, is also linked to stress, is associated with Alzheimer's disease as well. So this statistic is pretty sobering. 40% of Alzheimer's patients suffer from severe depression. 21% of people who have depressive symptoms that increase over time were eventually diagnosed with dementia. So given this impact of stress on, our mental health, also the progression of Alzheimer's disease and potentially other degenerative diseases, really devastating diseases in our society, my lab has become really interested in how chronic stress is changing the brain at the molecular and cellular level. And I will tell you about some of our work on this. So just to give a little background to start with, as many of you probably know, the brain is made up of cells called neurons, and those are shown here. We have about 100 billion of these in our brains, so a very large number. Neurons communicate with one another through synapses, which are depicted as these little glowing spots here. And it's been estimated that each of our brains has actually more synapses than there are stars in the entire universe. I don't know if this is really accurate, but we have a staggering number of synapses, 10 to the 14th power. And anything that alters these connections will alter our thoughts and actions and can lead to brain disorders. We know that stress affects specific brain regions, and these are the hippocampus, which is important for learning and memory, the amygdala, which regulates our emotional arousal state, and the prefrontal cortex, which is an area that helps us to focus our attention and manage our impulses and emotions. And we know that exposure to glucocorticoids, the major stress hormones, actually alter the synapses, the connections in these brain regions. And so, for instance, in the hippocampus, glucocorticoid exposure causes a loss of synapses, and this leads to learning and memory problems. And so I'm going to show you, excuse me, what this loss of synapses looks like. So this is a picture that I think is really cool. There is a way that we can visualize neurons in the brains of mice using green fluorescent protein, which you can see here. And so these are all a bunch of labeled cells in an area of the hippocampus in this mouse. And if we zoom in on a couple of these neurons shown here, you can see these branched structures, which are called dendrites. And if we then were to zoom in on one of the dendrites, that's shown here, you can see these little protrusions coming off of the dendrite, and these are called spines. And each of these spines is actually a synapse. And under chronic stress, we know that these synapses are lost. And so you can see that here when you're comparing the spines in the animal that had chronic stress versus the control. And the loss of synapses in turn leads to the loss of these apical dendrites, which are shown here. So you can see that there are fewer of these dendrites in the neuron that was exposed to stress versus in the control neuron. And this in turn leads to less connectivity between the neurons. When you think about the whole area and all of the dendrites and all of the connections being made between neurons, there's a loss of this connectivity, which then causes learning and memory problems. And so a question you might be wondering about is, how does stress cause this loss of synapses? And so to explain this, I'll tell you just a little more about the structure of neurons. So as we saw in the last slide, neurons have these branched structures called dendrites that are depicted this way in the cartoon here. And they also have this long, thin process called an axon, which is here. And the dendrites actually are the ones that receive the input from other neurons, and the axons are the ones that are forming the connections onto dendrites of other neurons. So if we were to zoom in and look within an axon, we would see a protein called tau, which is this little sort of wiggly line here that is enriched in axons, found at high levels within axons, and is really important for maintaining the structure of axons. And normally it's at high levels, which is indicated by the red in the axon, and low levels in the dendrites. But then upon exposure to stress and glucocorticoids, tau levels actually go up. Tau accumulates in the neuron and starts moving into the dendrites. So if we were to zoom in again on a spine within the dendrite, which we'll show here, a little schematic of a spine, what we would see is that as this protein tau is accumulating, it moves into the spine, into the synapse, interacts with this receptor that's found on the surface of the synapse, and basically causes this receptor to become overactivated, leading to the death of the synapse, the loss of the synapse. And over time, as these individual synapses are lost, this leads to basically the retraction and loss of these dendrites. So to summarize what I've shown you so far, we know that stress causes accumulation of this protein tau in neurons. This then causes synapse and dendrite loss, and that in turn leads to learning and memory impairment. So then what my lab got really interested in is this question of, how is stress causing accumulation of this protein tau? And so to look at this, we collaborated with Yannis Soteropoulos, who's an investigator at the University of Minho in Portugal. So we have really complementary interests, because my lab is interested in sort of cells and proteins, and his lab is interested in animal models of stress. So we have a really nice kind of synergistic collaboration. And I won't go into the details of our study, but basically we uncovered a pathway for tau disposal and found that this was under the control of glucocorticoids, these stress hormones. So to just use this analogy of garbage disposal, tau is normally delivered kind of through a garbage, through a garbage truck to the waste disposal plant in the cell. And there is a sanitation worker who puts tau into the garbage truck. And we found that in neurons, this sanitation worker is actually a protein called Rab35. And we also found that glucocorticoids essentially decrease the number of these Rab35 sanitation workers in the neurons. And so this leads to the accumulation of tau, because now there's nobody to sort of take tau to the garbage truck, as it were. And so we wondered, if we put a bunch of these Rab35 sanitation workers into the neurons, would this help to protect the brain against stress? So we did an experiment that I'm going to kind of walk you through to test this. And so what we did was we injected this Rab35 kind of sanitation worker into, we injected a virus to express this protein into the hippocampus of rats, or we also used a control protein, which is just green fluorescent protein, which doesn't cause anything to happen. Then we waited a few weeks and then injected the animals either with a saline control or with dexamethasone, which is a synthetic glucocorticoid, for 15 days to basically mimic stress. Then we examined the neurons in the hippocampus of these animals. And as I'll show you here what we saw, which was pretty profound. So in the control animals, these are animals just expressing GFP protein. You can see that there is this loss of dendrites in the animals that were injected with glucocorticoids. So they have fewer dendrites than the control animals. But in the animals that were expressing Rab35 in the hippocampus, we see that there is actually no loss of these dendrites in response to the glucocorticoid injections. And this is quantification basically showing this, that the expression of Rab35 protected these animals against the loss of dendrites and also the loss of spines on the dendrites. So we were pretty excited about this finding because it really showed that the expression of this sanitation worker in the neurons was basically protective against the effects of stress and glucocorticoids. And to be clear, we're not advocating injecting people's brains with Rab35 by any means. I don't think that would be a good idea. But this was a proof of concept to us that basically by turning up this kind of garbage disposal pathway, we could stimulate the removal of this protein tau, and this would actually prevent the effects of stress in the brain. And actually, this is also, as it turns out, highly relevant for Alzheimer's disease. The two major kinds of pathology seen in the brains of Alzheimer's disease patients are basically amyloid plaques, which are shown here, and tau neurofibrillary tangles, which are shown here as these kind of dark, this dark coloring in neurons. And neurofibrillary tangles are essentially accumulated tau protein that can't be disposed of properly. And we know from research over about the past 10 to 15 years that tau is actually a major driver of Alzheimer's disease progression. So the accumulation of tau in neurons is toxic, partly due to the same mechanism of synapse loss that I talked about a couple of slides ago, and partly because these aggregates of tau can actually be passed from one neuron to another. And this actually spreads tau pathology sort of through the brain and drives the progression of Alzheimer's disease. And so based on our findings and those of other labs, we think that tau is really a driver of this vicious cycle of stress and Alzheimer's disease. And so we know that increased levels of tau are actually driving the loss of synapses, leading to the loss of synapses, and also causing other problems in the brain like inflammation. And these things together are triggering depressive disorders. Depressive disorders are also driving further Alzheimer's progression. So there's a whole cycle happening where tau is kind of one of the instigating factors. And there have actually been a number of recent efforts, both in academic labs and also in the pharmaceutical industry, to target tau for the treatment of Alzheimer's disease. And in particular, tau reduction strategies are being used. So things that reduce the expression of tau, or also drugs that are targeting this, what this is referring to here are basically the garbage disposal pathways that we were discussing. So ways of causing tau to be disposed of and ways of preventing high expression of tau in the first place are things that are being used for in early stage Alzheimer's disease testing. And these are some of the drugs that are currently in clinical trials. You can see most of them are in pretty early stages, but a few of them actually are advancing. So I know I kind of covered a lot of ground pretty quickly here, but here are just sort of the main take-home messages of the presentation. So I have shown you that, or told you that chronic stress can damage the brain. And we know that this can cause a number of different types of disorders like depression, anxiety, Alzheimer's disease. There's evidence also that stress can stimulate the progression of other degenerative disorders as well. We know that one cause of this damage is accumulation of this tau protein in neurons and in its synapses. And then finally, therapies that reduce tau levels, we've seen, in our hands and also in other labs, there's a lot of evidence that indicates that these have promise for treating diseases, and they're currently in clinical trial for Alzheimer's disease, other neurodegenerative disorders that are driven by tau, such as frontotemporal dementia. And it's possible that they would, knowing what we know about stress and the links to depression and anxiety, that these treatments potentially could help for things like depression as well. And of course, in my completely unscientific, unprofessional opinion, stress reduction techniques are also great. And hopefully we have all learned some of these during the past year, which I think are also helpful in all aspects of our lives. And then with that, I just want to, last but not least, acknowledge the people in my lab who did the work, particularly my student Joao, who did this tau study, both things that we did in the molecular level and things that happened in Portugal at the animal level. So he was really a star student who did a lot of work on this project. And then I want to acknowledge my funding agencies, and in particular the Brain Research Foundation, who funded actually an earlier project in the lab that really sort of laid the groundwork for our current work, really got us thinking about, garbage disposal in the brain and how this process is important and how it might be relevant for disease, and, really kind of took us in exciting directions that we had not anticipated when we started the work. But they gave me funding when I was, just starting my lab and basically just getting going. And that's, I think, a really important time to be getting these, these little infusions that help you to kind of start risky projects that you might not otherwise think about doing. So really want to thank the foundation and the family who gave me the named grant for being able to do this work. So thank you very much, and I'm happy to take questions.
So thank you so much, Clarissa. I think this was really interesting. I have some really quick questions, but also if you connected because of the Zoom link, you can type some questions in chat. But as we're waiting, if somebody has something to ask, I'm going to ask a couple. Well, first of all, I'm happy that BRF helped you think about garbage and garbage disposal. So, and that you were able to branch out from the Sea Grant that we gave you and made some really great progress. I had a question though about chronic stress. How long, these are in rodents, the experiments. How long is chronic stress? Is it a month, a week, a year, and then in rats and then in humans?
Yeah, that's a really good question. In animal models, it's usually on the order of 2 to 4 weeks. And interestingly, a lot of in the animal models, a lot of these effects are reversible over time. So if you were to then, stop the stress, and, stress in animals is basically things like cage crowding, and it's a lot of things that maybe we would also just find to be stressful in humans, but not something really extreme, something that is sort of equivalent to mild stress. But it doesn't take that much. Mild stress for several weeks is still, chronic. And the effects are generally reversible in the animals. And I think, I'm not sure one could easily, with animal approval and all that stuff, kind of know how long you would do the stress to have it be irreversible, or when that happens or anything. And I think in humans, when they have done epidemiological studies showing these links between stressful experiences in life and later Alzheimer's disease, for instance, it's usually sort of a couple of years, is kind of what people will report. Or, it gets a little complicated because some things are quite chronic, like people who suffer from depression, that can be sort of on and off for years. But sometimes it's as little as a couple of years of some sort of very psychologically stressful event that can sort of predispose people for, later life degenerative disease. So it's always a little sobering for me, maybe especially over the last year, to see these studies. But I think it's also so individually dependent that, people who are more resilient, people who are more susceptible. So it's very, these animal models, they're very, the backgrounds are very well controlled. And obviously in humans, we don't have well controlled backgrounds. So there's a lot of factors, I think, that go into it. But
well, this kind of leads me to another question. What about people that are suffering from post-traumatic stress disorder? Are they at a higher risk for Alzheimer's?
That's a really good question, and I don't know if PTSD is a somewhat recent diagnosis, and I don't know if they have had a chance to do the really longitudinal studies that one would need to completely be able to verify that at this point. But certainly in say veterans of wars who were known to have suffered World War I or II, there were higher rates of dementia and things, I believe, in some of these populations. Of course, then you have other issues like chronic blasts and things that sort of shake the brain and cause chronic traumatic brain injury that are also linked to Alzheimer's disease. So I think it can be a little hard to disentangle some of these kinds of things, but I'm sure that those studies are being done. And we have a lot of experiments that are inadvertent, like COVID pandemic, for instance, that might, end up leading to down the road a lot more information, unfortunately, about these links. So,
Clarissa, I had a question.
Yeah.
It's an excellent presentation on a very powerful subject, and I wonder, have you now stayed involved in Alzheimer's? And is that what you do, is take the next step beyond having completed this project? In other words, I see a finite project and it's excellent. Have you then gone on to new projects in the same area, or do you move on to something else?
Yeah, we're still looking at this. Yeah, that's a good question. We are doing kind of further investigation in this area, looking even more at some of what I was talking about that has to do with tau propagation. So sort of the moving of these tau aggregates between neurons. And we're kind of trying to understand how that is being driven by stress, because it looks like it is, in addition to the sort of disposal of tau within the cell, how it is that neurons are passing these aggregates from one neuron to the next, and how this might also end up being driven by glucocorticoids and stress. And so we're kind of building on our previous work and trying to do more. I'm much more of a molecular and cellular focused person, and my background is really in cell biology of neurons. And so I'm very interested at the level where you can look in individual neurons and see, how do these hormones impact these molecular events that are happening within the neuron to cause, a lot of things to happen. So there is a lot of detail here that I didn't, want to go into relating to specific pathways and ways that things are being regulated that I think are still really interesting and will really be cool for us to work out. And then, we're continuing this collaboration with Soteropoulos group because they've been great and we've had a lot of fun with it, and they're much more at the animal level, and we're trying to, think about ways of doing more human-related studies and seeing if we can look for different types of biomarkers that might be able to tell us, about susceptibility and, people's susceptibility to stress and when that could potentially lead to Alzheimer's disease or not. That's something we're interested in. It's hard to do this stuff in humans because really what you want is a group of samples collected over many years so that you can kind of retrospectively look back at people and say, okay, they started here and now they have, this disease or they don't. And, what was different about these individuals over time or, things happening in their brains over time. So we're looking at a lot of these kinds of questions.
Thank you.
Yeah, thanks.
And then I'm going to ask you to look into your crystal ball. You told us that there are some therapeutic targets that they're working on, tau, that and a lot of them are still in the early stages, but some are moving forward. How promising are they? Do you feel pretty hopeful about some of these, and if you do feel hopeful, what do you think would be a timing? How long is this going to be in the pipeline before it becomes something that would actually be therapeutic?
Yeah, I think that definitely there are treatments that look promising, and I guess one of the types of treatments that I think has gotten a lot of traction lately are these things called antisense oligonucleotides, which are ways of basically reducing the expression of tau and other proteins. Those have been used to treat some other diseases, one of which is this one called SMA, which is a disease that affects children, movement disorder. I'm sure you know this, Terry, but yeah. And that's actually, those treatments have been quite promising in young children. And I think that has maybe pushed, that technology kind of to the forefront. And it might be that will be a reasonable strategy for treating some of these things. But, to be honest, we still don't really know everything about the normal function of tau. It's in neurons. It's a very complicated protein that has a lot of different things going on with it. So it's not really clear that you want to dramatically reduce its expression unless you have a compelling reason to do that. So I think a lot of these treatments will sort of have to do with the timing. And, we know from a lot of other studies in Alzheimer's disease to do with amyloid clearance, for instance, that a lot of times probably we're sort of treating too late before, when you can't really reverse processes. So I think there's this really tricky balance of getting a treatment into somebody at a time point where you can prevent things from happening, but yet not be messing around too much with the biology of the system and of the protein and what tau it normally has to do in the cell. You don't want to stop that from happening either. So I think unfortunately with brain diseases, we still have a ways to go with therapies. And I think we're just starting to see some really exciting ones, some really neat, progress, which hopefully will kind of continue to fuel more discoveries and more good treatments. But I think it does require a little patience because it's sort of the organ we least understand at this point. So, yeah.
Well, I mean, I think that that's a perfect segue for me to say, we've been around since 1953, so we know that it's a long haul, and we are discovering new things. It just takes some time. Emily had a question. Do SSRIs, or you can also see these, right?
Let's see, I guess I probably can if I can look at the chat.
Antidepressants impact the release of
Yeah.
tau proteins.
That is a really good question. I don't know that. I might need to, I'll have to look that up maybe and see if, whoops, sorry, if anything is known about that. Certainly treating depression, I think in any case is going to be a good strategy for preventing, the progression of these diseases. Whether there's any direct link to tau with the SSRIs, I'm not really sure about. But yeah, that's a great question.
All right, well, it is 6:31, so I'm going to say again, thank you so much, Clarissa, for this interesting talk. And I'm so happy that, we were able to give you a grant, support your early research, and you've certainly taken off, and congratulations on your moving up and getting tenure. I watched your talk on YouTube. So again, thank you, and thank everyone for joining. I know I learned a lot, and I hope everyone else did as well.
Thank you.